All right, class. So now we are going to talk about different ways to produce muscle tension. We will focus on the graph. And if you look at the graph on the x-axis, there is time in milliseconds. And on the y-axis, we have tension or the force of muscle contraction. Now on the x-axis, we also see some orange arrows. Those are stimuli, singular stimulus. And with time, we do notice that we increase the frequency of stimuli. Okay? All right. So let's focus on the very first graph thing here. This is called a twitch. A twitch is caused when a single action potential causes a muscle to contract, and then the muscle contracts and then completely relaxes. So there is contraction followed by full relaxation. That's a muscle twitch. If you look at a twitch, it has three phases. The first phase is called latent phase or the latent period, where the action potential travels down the sarcolemma, but the contraction has not started yet. Then we have the actual contraction and then the relaxation phase. Okay, so that's a twitch. Now, hold on just a minute. Okay, then we have summation. Summation is when, let's say there is a twitch, but the muscle has not relaxed yet. So if a second electric shock is delivered before the muscle has had a chance to fully relax from the first twitch, the second twitch will kind of piggyback on the first, and this is called summation. So summation is when the frequency increases. So right here we see there are three different stimuli. So there is obviously increase in frequency of the stimuli, and then before a twitch can actually relax, there is another one and another one. So full relaxation cannot take place, and the second one kind of rides on the first one, and the third one rides on the second one, and this is what summation is. Now if we further increase the frequency as we see here, then this leads to incomplete tetanus, and this is where the maximum tension is reached. So this is maximum tension. However, there is a little bit of relaxation time between successive twitches, and this relaxation time gets shorter and shorter, and this is incomplete tetanus. And then complete tetanus is sustained contraction in the smooth muscle. So this is maximum tension, of course, but there is zero relaxation between twitches. So that is sustained contraction leading to complete tetanus. If tetanus continues for some time, that leads to fatigue state of the muscle. Okay, now based on this that we just talked about, there is a stimulator experiment that you can get to do. But basically this experiment is performed to elicit muscle contraction using electrical impulses. Here we have a student, and the student has electrodes set up against his body, and the electrical impulses, they actually work as action potential that will run down the muscle fiber, run down the motor neuron, ultimately leading to contraction of muscle. And here the goal is to achieve finger flexion. So the goal is to see at which voltage and at which frequency the student gets finger flexion. So this is the instrument that is used in the lab, and the instrument here has okay, so this instrument has 3 knobs that I want you to look at, and also you will see there are multipliers associated with each knob. The first knob says events per second. That means it's measuring frequency. It is actually measuring how often you are being shocked. Okay, and associated with it is a multiplier, so you can do it like 2 times per second, you can do it 20 times per second, or 200 times per second. The second one is duration, which is in milliseconds, and that actually tells us the length of the shock that we will experience. Okay, so it can be 10 milliseconds, 50 milliseconds, and then also it has a multiplier. And the third one is amplitude, which is actually showing us the strength of the shock. And I want you to think of how will the strength of a shock affect the force of muscle contraction. So if we change any of these, if we change the frequency, if we change the duration, or if we change the amplitude, how is that going to affect the muscle contraction? I will post a class discussion board. I'll open up a class discussion board, and I want you to think about these scenarios and talk to your classmates about it, because most definitely you will get questions related to this on your practical exam. Also, there is a YouTube video that I have posted associated with it. It's on the PowerPoint, so please go and review that. So yeah, this is a YouTube video that I was talking about, and they do a similar experiment that we do in lab, but not the exact one. And here, so the experiment, in this experiment, they try to find the initial threshold, and then they keep the duration same. Now, for the twitch, obviously you have the single pulse, but then as you go to a different voltage, I want you to record the observation. So for this chart, I want students to go to the YouTube link, which is right here. So go through the video and fill out the observation here. Okay. One thing I would like to point out, look at the frequency. For summation, obviously, the frequency is increasing. And for tetanus, incomplete and complete tetanus, the frequency is way more. So keep all these things in mind. Look up the YouTube video and write down the observations here. Okay, moving on to stretch reflex. So to start with, let's talk about reflex action first. Know that a reflex is a quick involuntary response to a stimulus. It does not need involvement of higher order processing centers of the brain. So your spinal cord can take care of reflexes. In this lab, we will focus on muscle stretch reflex, which carries sensory information from one part of the body and sends motor command to the same side of the body. So in this diagram at the bottom of the page, we see that the hand is touching a hot object, and when we touch a hot object, we immediately withdraw our hands, right? So there are sensory neurons in red that carry that information towards the spinal cord. Towards the spinal cord. There are interneurons that decide what action to take, and then it sends out a motor command via the motor neuron that comes to the muscles here and tells the muscles to contract and withdraw the hand. So this is a reflex pathway or a reflex arc. Okay, now when skeletal muscles are rapidly stretched, the muscle stretch reflex will cause it to contract very quickly, and this is actually done as a protective response, so the muscle is not damaged. This diagram is showing a knee jerk reflex, very similar. Actually, this is the one that health professionals test on their patients. So the stimulus here is a rubber hammer. With the rubber hammer, you tap the tendon of the quadriceps muscle, and as you tap, that is sensed by these receptors which are present on your quads muscle. That carries, so these are the receptors in blue. The receptors then send the information to the spinal cord, and the spinal cord then to the spinal cord, and then the sensory neuron communicates with the motor neuron in red, and the motor neuron brings out a command telling this muscle, telling the quadriceps to contract. So stimulus, sensors here. This is the afferent pathway carrying information to the spinal cord, and then the motor neuron brings out a motor command to the same muscle, causing it to contract. So this is the afferent pathway, this is the efferent pathway. However, I want you to follow that the sensory neuron or the somatosensory neuron also coordinates with an interneuron here, and the interneuron then sends out a command to a motor neuron that innervates the hamstring muscles, and this is actually a negative and inhibitory stimulus. So this is positive, but this one is negative because this is causing the hamstrings to relax. So as the quads contract, the hamstrings relax, and that leads to the extension of your feet, and you see the knee jerk reflex in the patient. On here, you see the blue, red, and green. They represent sensor, effector, and controller. So there are 2 YouTube videos that are posted. They're really good. They talk about jaw, biceps, triceps, and ankle reflexes. Now I want you to know that the adult Babinski reflex is different from the infant Babinski reflex, and when you watch these videos you will have a better understanding of these reflexes. Now in the lab we use a rubber hammer, but since you are sitting at home, if you can find something similar to this, use it on a family member and see the results. You can follow part A of your manual and continue the experiment. However, make sure to maintain social distance and wash your hands if you are doing it on any test subject at home. All right, so that's it for today. Okay, thank you, and this was the last video. Next week we will talk about sensory lab, and I will post videos associated with sensory labs as well. Thanks.